Yep.
Reactive oxygen species generation simulants. Okay.
Okay, very good.
Exactly. Chairman, we don't want to keep time here.
Thank you. Yeah, I put this picture up of the one geologist that actually explored the moon, and you can see if you're sitting up front that his spacesuit is covered in dust. And for those that might explore the moon for a longer period of time, it's going to be very difficult to avoid some exposure to dust, and that's really what drives most of our research. And so this is done as part of the RISE effort. And now let me see. Okay, and where are the little at the bottom? I don't see any. Okay.
Hidden so well. Oh, we have to press this button first.
Okay.
And now you
Okay,
can go.
very good. All right. So in terms of exposures in an environment like that, the one thing that we're mostly concerned about is exposure through inhalation. And just to give you a sense of how important this interface is, it is the same if you lay out the surface area of the lung where it exchanges air with the bloodstream, which happens to be composed of very small little sacs, if you will. If you lay that area out, it's the same area, surface area, as the playing surface of a tennis court, just to give you a sense of how large this interface is. Now, if we are confronted, if the human lung is confronted with foreign material, the immune system starts to kick in. And the immune system is essentially a broad spectrum defense system, and it relies on producing hydrogen peroxide, or bleach, if you will, and that then starts to react. If this foreign material happens to be minerals, it may react with these minerals and actually form radicals, and radicals are not good for you. They damage proteins, DNA, and also cause, in general, just stress in cells. And so if you are exposed to particles on a regular basis, then this can lead to chronic inflammation. That sets you off on a path of diminished health. It can actually lead to lung cancer, and asbestosis is another kind of disease in that category. So the other response might be that the body tries to encapsulate these particles, and then it causes diminished lung function because you get fibrotic material growing around it, and it essentially takes lung capacity away. So if you look at the chemistry, I'm a geochemist by training, so I'm interested in the chemistry of this. If this is your a generic dust particle, there's a few different things that this dust particle can represent in terms of reactivity. First is defects, and we talked a little bit about defects earlier. This is unsatisfied bonds that will react with the medium. There could be oxygen, CO2, water can be involved, and that leads to a variety of different radicals, either carbon-based or oxygen-based radicals. We can also have iron, for example, dissolve into cell fluid or lung fluid, and this can, and as well as other metallic elements. But here's a reaction that becomes important. That is the Fenton reaction, where we have ferrous iron reacting with hydrogen peroxide and then leading to ferric iron, but most importantly, the OH radical is formed. Now, where does this hydrogen peroxide come from? Well, the hydrogen peroxide can actually be formed through two different independent mechanisms. One is that we have oxygen in the fluid, because we live in an environment where we're going to be exposed to oxygen, otherwise we won't live at all. And so the oxygen will actually stepwise be reduced, and one of the steps is the production of hydrogen peroxide. So that's one source of hydrogen peroxide. The other thing is, as I mentioned before, the immune system actually has enzymatic processes that can lead to hydrogen peroxide. So both the oxygen reaction and also the enzymatic reaction can lead to hydrogen peroxide. So we have the ingredients of iron, ferrous iron, and hydrogen peroxide, and then this can drive the formation of OH radical. Now, how this plays out in the cell is that it can lead to chronic inflammation, mutagenesis, and essentially this here is cell death. So we have a variety of different outcomes that play out on the cellular level. Okay, so what we're interested in the RISE team is that we try to understand how this might play out on over time. And the first thing that we want to do is simulating materials on airless planetary bodies. And so here are some of the factors that we are trying to resolve. There's composition, there's mechanical stress, the change of the surface by removing water or OH groups on the surface, so dehydroxylation, and then also this much more complex issue of space weathering. So what we have in terms of research strategies is to have a number of different materials that we can start with that actually relate to composition. So we can look at simulants that are available, synthetic materials that might be produced, and also we can look at single minerals that are major minerals in the materials that we're interested in. We can simulate mechanical stress by grinding. We can simulate this dehydroxylation by heating, and particularly heating in vacuum. And then the space weathering, we're actually in a position to leverage facilities at Brookhaven National Lab where we can actually implant protons, and we also can expose materials to UV and IR lasers. And so that then is a proxy for space weathering. And the goal here is to prepare well-characterized materials for reactivity, biopersistence. But biopersistence is really how long a particle might actually reside or stay in the lung. So, for example, asbestos particles many asbestos materials are very insoluble, so they persist long in the lung, whereas something like a salt crystal will dissolve immediately in the lung fluid. So that's one important component. And then we are working together with a team in the health sciences where we actually do exposure studies. And let me take a look at what we do there. So on the impact on human explorers over time, we really distinguish a variety of different sort of categories of things that can happen to you that play out over different time scales. So we have this oxidative stress that leads to inflammation. And this is really the idea that, for example, we can have hydrogen peroxide formed due to the minerals reacting in lung fluid or in cell fluids that lead to this hydrogen peroxide generated by the minerals. And so we have a variety of different strategies where we can exploit this, including also in epithelial cells. And this is a particular strain of epithelial cells that we've worked with over the last 10 years or so. We can look at cell damage and death, and we, for example, can measure protein damage in these epithelial cells. And we also more recently have been starting to use tissue that is harvested from mouse lungs as a model. And then the next level of injury, if you will, is genotoxicity, which may actually play out over a much longer time scale. So this may be, for example, years after somebody might come back from exposure. And that can be triggered by DNA damage, and we can actually measure that. There's also the inhibition of DNA repair. So it's really a balance between how much damage there is. Your body is constantly damaged, but it's also constantly repairing it. So you can have damage resulting by inhibition of the repair system. And then finally, fibrosis, which really is outside of our scope of study, but this would actually involve animal studies. For example, you would take mice or rats and expose them to the material. So this slide here kind of gives you a brief overview where we are in terms of the composition of our team and what we actually do. So I'm a geochemist, as I mentioned. I'm merely interested in the reactivity of mineral surfaces. So my lab is specialized at looking at the production of these what we call reactive oxygen species, which are primarily hydrogen peroxide and OH radical. And we can do this by dispersing these materials in water. We've also done experiments and will do experiments in simulated lung fluids. Then we have an effort that looks at specific mineral, in particular olivine. And there's actually a poster here from Joel Hurwitz's lab, Sarah Port's work. And then we look at the effect of exposure to simulants in mice lung tissue, and that is a poster by Jill from Circa Lab. And then we have also a poster, and Rachel actually introduced it this morning, on genotoxicity. So there's a spectrum of different studies, but they all hang together. So what we do in my lab is, what we're going to talk about today here to finish up my talk is specifically looking at hydrogen peroxide generation. And the way we do this is we actually use a technique that is developed for the medical world, or biomedical world, where we have a small probe. You can't really see, there's a small little probe that we can actually, that is designed to go into cells, but we actually can stick it into slurries of materials. So here's a close-up picture. So we can do these experiments in very small slurries, which is important because a lot of the material is not available in large quantities. So we disperse this material in water in these studies, and then we can do this with or without EDTA. Now, the EDTA is important because if we add EDTA, we actually arrest the Fenton reaction. So in other words, if the Fenton reaction goes forward, we can't measure the hydrogen peroxide anymore. So basically, we cut off the Fenton reaction, and we can measure the amount of hydrogen peroxide, which really is one of the key things that we are interested in. So because it's a probe, we actually can measure this hydrogen peroxide formation continuously. And that's actually a breakthrough because up to now, or until recently, we use molecular probes which give you an integrated concentration over time. So let's say you react something with a molecule, it converts the molecule, and you get one data point. Now we're getting actually a dynamic output of how hydrogen peroxide evolves over time. We've started to look at this mechanical treatment by simply because we have small amounts of material in many cases, so we just do hand crushing by doing this in air, but also under nitrogen in the glove bag, and that's what this picture represents. So here I'm going to show you just a few data points. We have actually a lot of data, but I'm going to share just two extreme sets of data. So this is probably the most used simulant. This is the JSC1A, and this is experiment done in air. So here we are taking this particular simulant, which actually we can get in large quantities. We measure the hydrogen peroxide using this probe, and this is time. So these experiments run for about 20 minutes, and what's important here, we have hundreds and hundreds of data points. So just to give you a sense of how this works, if we take this material and we take it out of the jar and we add the EDTA to it, we get this green curve here. And that's actually a modest amount of hydrogen peroxide. Then we can repeat it. We can, for example, crush it and immediately measure it. This is that light blue curve which goes up here, and it's slightly higher. Okay, and then we let this material sit for 1 day, 3 days, 9 days simply in air. And the reason that we do this is because we expect that the surface will start to react with the water vapor, with CO2 in the air, and so forth. And we see slight changes in reactivity over, like for example, here is the 9-day curve. It's still higher than the uncrushed material. Now, if we do this now in nitrogen, these data was collected this week, so we don't have as long a time frame. But basically, we have the fresh crushed material. We see a slightly higher amount, or about the same as last time, and then we have 2 days crushed. Is basically we recover the same curve. And the point here is that once we do this in nitrogen, we don't have the air anymore, we don't have the CO2 anymore, and the reactivity is essentially remains on the surface. Now, to go to another extreme is another simulant that we've received, where we actually can do this same experiment, but now we're starting to recover concentrations up well over 100 micromole of hydrogen peroxide. So that's more than a 10-fold increase. And to make a long story short, when we do this uncrushed, we actually recover nothing if we have no EDTA present. What that means is that it immediately reacts to become OH radical, and that's why we can't see the hydrogen peroxide. We start seeing it if we add EDTA, but if we then crush it, we actually get the highest numbers, and it decays over time. This is in air, and just to wrap up here, if we do this in nitrogen again, the reactivity is essentially locked into the surface. So the exposure, what you do, you can treat something, but the exposure afterwards becomes very important in terms of locking in the reactivity. Now, we've done a whole host of these simulants, and I'll just point out that those that actually have the highest glass content seems to be the most reactive. I've showed you data for this particular simulant here, which has 44% glass. Another one that produces a significant amount of hydrogen peroxide is this OB1. It's got about 43% glass. And in all cases, we see higher numbers if we do this under nitrogen than under air. The next step is to actually use a new instrument that we just got to actually measure OH radical, and we won't have time to talk about this, but that's the next step. And these are the people that have been working with me. And I would like to point out Doug Rickman, who's actually very important for us because he's been actually giving us some of these simulants, and some of these simulants are hard to come by. And we're looking forward to continue this research to drive this to a better understanding of how these materials could contribute to health effects. Thank you.
right, very nice talk. And of course, we do have time for questions if there are any. Okay, I have one question for you. When you crush it under a nitrogen bag, have you ever taken out and exposed it to air? Perhaps you showed that slide and then done
No.
that.
So when those data points it's actually maintained in nitrogen. Yeah. The idea is to maintain the nitrogen, but we can do all kinds of permutations at that point. Yes.
It'd be interesting to see how quickly it passivates in air.
Yep.
Yeah.
Absolutely. Yep.
Okay. Very nice.
Somebody left a cell phone here.
That'd be me.
Oh, okay.
Thank you.